I am told that we are ready and are live on the webcast. I am Catherine Page. I am the chair of the SICATM, and I also work for the Clorox company. We are going to go around the centre table first to introduce ourselves, just like we did yesterday, brief introductions, and then we'll proceed with the other SICATM members that are joining us online. Okay, I'm going to go to my left.
John Gordon, ICVAM member with CPSC.
Monique Perrin, US EPA.
Nathan Price, SICATM member. I'm a professor at the Buck Institute and Chief Science Officer at Thorne.
Patricia Silveira, SICATM member, professor at Indiana University School of Public Health.
Priyanka Sura, SICATM member, toxicologist at Gilead Sciences. Sally Thompson-Aratani, University of Washington.
Misty Ushio, SICATM member, managing partner at the Health Ventures.
Charles Benham, associate professor, University of Pittsburgh, SICATM member.
Jennifer Good, ICVAM member, US FDA, Center for Devices.
Barnett Ratner, ICVAM member, Department of the Interior.
Good morning. Steve Wong, Department of Transportation. Thank you.
Brian Chalua, ICVAM member, National Cancer Institute. Adrian Nania, SICATM member with Servier Pharmaceuticals.
Sue Marty, SICATM member from Dow Chemical.
Sue Leary, SICATM member, Alternatives Research and Development Foundation. Ellen Berg, SICATM member, Alpha Predict.
Antonio Baines, SICATM member, associate professor, North Carolina Central University.
Melanie Brownlow, NIEHS, also the designated federal officer for SICATM.
Good morning, Heather Patisal, scientific director, Division of Translational Toxicology, NIEHS.
Hello, Nicole Feinstreyer, NIEHS Division of Translational Toxicology, NICETM director, and ICVAM executive director.
Yes, I have the microphone. Can you hear me? There we go. Emily Ranke, Innovative Principal Investigator, Contract Reporting, NICETM. Rebecca Rosses, alternate SICATM member for U.S. Department of Transportation.
Liza Taye, Johns Hopkins University.
Amy Klippinger, PETA Science Consortium International.
Anton Simeonov, NCATS, NIH.
Kemal Mansouri, NICETM.
Chris McPherson, NIEHS, Division of Translational Toxicology.
AJ Newell, NIEHS, Division of Translational Toxicology.
Irene Pulvers, Food and Drug Administration. Helena Hoberg, Deputy Director of NICETM.
Stephen Moorfield, Innovative Program Manager supporting NICETM.
Kathy Sprankle, Innovative Communication Specialist supporting NICETM.
Tim Schaefer, Environmental Protection Agency, Office of Research and Development.
Can we proceed with introducing the other SICATM members that are online?
Hi, Stepan Baran, SICATM member, Veristim Life.
Hello, Corey Ellison, SICATM member, Procter & Gamble.
Good morning, Christina Miles, Head of Regulatory,
at the Honey Pot Company, also Cicada member and adjunct faculty at Spelman College.
Great, thank you. I'm going to take this time to acknowledge the representatives from the International Corporation on Alternative Test Methods, or ICATM, that are viewing the meeting remotely. And I also acknowledge that some representatives of the Interagency Coordinating Committee on the Validation of Alternative Methods, or ICVAM, are also present online. I would like to introduce our DFO, Melanie Brownlow, for some housekeeping and COI statement and ground.
Thank you. Good morning, everybody. Can you hear me okay? Thank you. Thank you. So starting with the conflict of interest statement, members of the Scientific Advisory Committee on Alternative Toxicological Methods, or SICADM, serve as individual scientists and not as representatives of any organization. Each member is to exercise judgment prior to or during the meeting as to whether a potential conflict of interest might exist relative to the topic under discussion due to his or her occupational affiliation, professional activity, or financial interests, or those of their spouse, minor child, partner, or organization. Members are asked to bring to the attention of the federal officer any potential conflict of interest that may pertain to a topic being considered at the meeting. Should there be a potential conflict of interest, it will be handled in accordance with departmental policies and requirements. So for housekeeping, very much like yesterday, this is a public meeting that's being broadcast to a remote audience. So when speaking, please remember to introduce yourself and state your affiliation. Additionally, since this is a public meeting, we ask panelists to reserve the use of the Zoom chat box to technical issues only. If having any technical issues, please message me. All discussion in this meeting will take place in the public forum and will be captured in the meeting minutes. And while we have a scheduled lunch for break, the actual time may vary and be at the discretion is at the discretion of the chair. Slides will be made available on the website as soon as they meet government requirements for accessibility, and minutes will be posted to the website and also after they've been reviewed for accuracy, approved by the chair, and meet government requirements for accessibility. Thanks, everybody.
Thank you, and we're going to start with our next topic of developmental neurotoxicity. This is going to, the day is going to pan out same as yesterday, so after each talk we will have some clarifying questions, and clarifying questions only. The discussion questions will be held until the end of each session. So I'd like to introduce Dr. Mangus, who is going to present remotely.
Hello, good morning. Thank you, Madam Chair. Hope you can hear me well. I'm gonna share my screen, but I think somebody needs to give me permission. Okay. You should be able to see my screen now. So it is my pleasure to be the first speaker of this focus session on developmental neurotoxicity. I'm a regulatory toxicologist working in the pesticides peer review unit of the European Food Safety Authority. And I'm currently also co-chair of the OECD DNT in vitro battery expert group. So in this presentation, I'm going to focus on the efforts at the OECD at EFSA level, not because they are more relevant, but because my colleagues from US EPA, DTT, FDA will present after me. Allow me to start from the beginning by saying that assess for developmental neurotoxicity is challenging in the sense that we are in a complex endpoint. A DNT chemical is defined by a very diverse set of substances that interfere with the normal development of the nervous system, which, if perturbed without compensation, may lead to diverse adverse effects on the structure or functions. So we are talking of a complex substance that has quite heterogeneity in terms of cells, molecules, and biology. In addition, there are many molecular initiated events or molecular targets that are not well understood, and the full pathways of adversity or of compensation are not well understood. When this happens, there are very diverse adverse outcomes that in human are divided by intellectual disabilities, language disability, tic or motor disorders, attention deficit hyperactive disorder, and a series of neurodevelopmental disease, which are quite different and that they are not happening in rodents, for example. So the timeline is also very different between humans and rodents. So I think we have to acknowledge the success in a way of the OECD expert group that in a short timeline has been able to develop an OECD approved by WNT document, initial guidance document that can be useful and is used currently by us, by regulators. And I think the key success factors has been the collaborative effort and also the use of the data in real case studies. And I'm going to present some examples of this. So starting from the beginning, the DNT in vitro battery was piloted by the advancement of science, obviously, but the advancement in using of the in vitro neural models, combining with high technology for developing the assays. But I think it's critical that from the beginning, a series of workshops and of meetings were organized that put it together the researchers, the developers of the assays, the mechanistic toxicologists, and the regulators or decision makers. So for us, for EFSA, we joined this effort and the history started for a regulatory need. I think it's the same in US, but in the pesticide regulation, there is a need of conducted a literature review of all the available evidence and include it in the regulatory dossier. So time ago, in 2013, we already had in vitro methods for assessing developmental neurotoxicity that we were not able to assess the robustness, the validity, or the reliability because there were not available guidance. So we joined the efforts, first by participating in a systematic literature review of the available methods after the readiness criteria. And with all of this process, we gained a better understanding. We also submitted the standard procedure submission for the development of the OECD guidance in 2017, and since then we are working with OECD. You have here a series of documents published by EFSA, in short, presenting the experimental work, different IATA case studies, and also the discussions that we had with our European stakeholders. The main milestone has been, of course, the approval by WNT of the OECD initial recommendation document that I'm going to present to you what contains and what does not contain. When we presented this document to our stakeholders, not only for the pesticides areas, but also for the chemicals area in general in Europe, there were an uncertainty analysis, let's say, and we launched a series of projects for further implementation of the DNT in vitro battery. I have to say that for EFSA, the question was never, can the NAMs or can the DNT in vitro battery completely replace the DNT assessment for pesticides? Our question was, can the DNT in vitro battery be helpful for refining, of course, reducing the animal testing, but mainly for assessing faster and assessing better for DNT? So this is the OECD initial recommendation document approved by WNT. It is actually, it has been shared by this committee, and I'm sure you already have knowledge of what contains, but just very briefly, it is quite a short document of less of 50 pages with a lot of appendices that contains the main documentation of the document. In the first part of the document, the main aim is to provide guidance in what the DNT in vitro battery is, what is the biology for us regulators with less experience, and what is the relevance of the DNT in vitro battery for assessing for DNT. There is a second part with a context. The DNT in vitro battery has been used in different contexts of use, and there are various examples as IATA, AOP inform IATA, or IATA. You will see today some of them. And then the most important part of the OECD document is the description of the essays of the DNT in vitro battery in the Appendix B, excuse me, where following the TOX-10, the full assay description of the 17 essays is included. There is another section on weight of evidence and how to integrate the evidence with the rest of the evidence. So in short, and I think this document, at least for us in EFSA and in Europe, is quite pioneering in the sense that the OECD guidance, it contains a battery, it's based on a battery of in vitro methods. Which ones? The one that were judged as ready by different international working groups, the ones that have tested a list of substances, of 81 substances, and the ones where we had the TOX-10, so the expanded version of the documentation of OECD guidance 211. The second main concept, and also is pioneer in my view, is the use of key neurodevelopmental processes. So the design of the battery for processes and not molecular or full pathways or molecular initiated events. And the use of AOPs as a framework for integrating the in vitro data. I don't know. Can you hear me well?
Yeah, we can hear you.
Thank you. Sorry, I had an audio issue. Excuse me. And the third main concept is the IATA as a tool to integrate the results of the DNT in vitro battery as a tool of doing the uncertainty analysis and as a tool to integrate it with other lines of evidence. I will present you how we do this and how we are currently doing almost in our business as usual in the pesticides area in Europe. I think it's important to note that the document the aim is to present the battery and to present how to evaluate the in vitro data. So what is a hit versus not hit versus a specific hit or not a specific hit, and what is the biology of the battery. It is not intended to provide interpretative guidance for a specific context of use or problem formulation, since this is quite regulatory dependent. So this figure from the OECD document, you can see all the essays of the DNT in vitro battery, the 17 essays. And as I say, they are organized by key neurodevelopmental process. So proliferation, apoptosis, migration, differentiation, neurity of growth, neurity maturation, synaptogenesis, glial differentiation, myelination, and neuronal network formation. It's important to know that some of them have human test system, others rat test system, and that there are already some future assays, some of them under development, other to be developed, that are summarized in the document. And there are two main regulatory assumptions when using this data is that the change is in this key neurodevelopmental process will be the result or will reflect the results of interaction with a series or with multiple molecular initiated events. And the second one is that if the target is achieved, what we observe in vitro, it would be also observed in vivo. So I think it's important for understanding the DNT in vitro battery to take into account that the brain development is high complex and there are very different temporal windows, and that we have different, and that this is also different in a species, but it's also different in the different areas of the brain. So we have here an example of the prefrontal cortex, but when these key neurodevelopmental processes happen, it's not the same in rodents that can be days versus humans that can last weeks or months. And they are not exactly the same. These temporal windows are not the same in the different areas of the brain. So here we have in the figure how the test system of the DNT in vitro battery represents the different developmental periods. Some of them represent more the embryonic or more the fetal period. The most important and also the longest section of the document is the elements for the weight of evidence. So what it has to be considered in terms of uncertainty analysis by us, by regulators, when including the DNT in vitro battery for decision making. Some are issues that are generic for all in vitro assays, but in a way for some areas, the integration for a systemic toxicity endpoint of in vitro method, it is a pioneer also by the DNT in vitro battery. So they are also included in the document, as can be, for example, the metabolic competence uncertainty, the applicability domain, in the sense of solubility of the substances that can be tested, or that the volatiles cannot be, just as an example, and the uncertainty of using nominal concentration and no intracellular concentrations. So all of this is well explained in the document, as probably you have seen. And then other important issues as the transparency in the data reporting and analysis, how to interpret individual assay versus how to interpret the full battery, and a series of advices for doing the uncertainty analysis together with the case studies. All of this, I think, at least from my point of view as regulator, can be summarized as the need of treat the data of the DNT in vitro battery as we do it with the rest of the data for a weight of evidence in the hazard assessment. And I'm going to show you what I mean for this. So for EFSA, as I say, the DNT in vitro battery is currently included when available in the dossier of the pesticides. And this is because we started the acceptability or the implementation project already in 2019 with the nomination of an EFSA panel IATA working group. So a series of experts as a committee, as we can say, also with our stakeholders that are ECA, member states, industry, GRC, or NGOs. So far we have published 3 IATA case studies. All of them follow the same workflow that is in line with the pesticides regulation. This series of IATA, AOP-informed IATA case studies have been endorsed by different regulatory bodies, as you have here in the slide. So from the Working Party for Hazard Assessment of OECD, but also our member states pesticides peer review meeting that are who have the decision in Europe for pesticides, or by European Commission, which are our risk managers, or by our pesticides panel, plant protection panel. So by a series of regulatory bodies that we have in Europe. This has meant that the DNT in vitro battery currently, if available, is part of our dossiers. So this is the AOP for IATA workflow, and it's nothing that we have invented. It's just how we conduct the weight of evidence of the data for assessing data for pesticides risk assessment. So it is based on systematic literature review. We can say evidence-based in the sense that we do, or a systematic literature review is done, and the data quality in terms of reliability, relevance, and reporting is assessed using a critical appraisal tool. Currently, we use a modified version of the OHAT NTP tool that we have fine-tuned for having specificities for the developmental neurotoxicity endpoints, measured in vivo, in rodent studies, but also in human observational studies, or in different in vitro or NAMs. We do not include so far in silico studies, but it's something that could be done. And also the critical appraisal tool will allow you to develop with a group of experts a critical appraisal tool for that. So this is in line, as I say, with the pesticides regulation, in the sense that in our regulation it's quite clear that we need to integrate all the available evidence and not only the OECD test guideline studies. Then the peculiarity we can say is that the data is integrated, is organized using an AOP framework. So the different in vivo, in vitro, or human endpoints are classified by categories of potential molecular initiated event, key events, or adverse outcome, depending on what is the endpoint that they are measured, and if we are talking of molecular or cellular or organism response. And then, of course, that data gap and uncertainty analysis, that is nothing new, is in line with the weight of evidence guidance, and is just to assess the reliability, the reporting, and the toxicological relevance of the observations. The conclusion, instead of doing a weight of evidence for one adverse outcome or for a molecular initiated event, the weight of evidence used the full body of evidence, so the AOP informed IATA. So these are our current methods for integrating the DNT in vitro battery, and this is what is part of the OECD document and what we presented to our stakeholders. And I have to say that when we have presented this, or also from the commenting phase of WNT, we can say only some keywords of the comments we're receiving. For sure, we will have the word validation of the need of optimization of the SOPs and the lab-to-lab transferability, the need of having tiered approach or approaches depending on the context of use, and further interpretative guidance, here again, depending on the context of use, the need of having an international agri-DNT reference chemical list. That is something easy to say, not easy to do. The need of having guidance and EBIF method, but specific for PBKs for DNT, and some commenting on the biological uncertainties. So the work that we are doing at EFSA, but also at the OECD level, is in that line. Here, just as an example, EFSA will publish soon a work that has been done by Kevin Crofton and Bill Mundy with a summary of all the in vivo DNT in vivo studies with the assessment that exists, but the number is only 164. So we know this doesn't cover the full chemical space. Another work that is ongoing is to have an agri analytical pipeline, and this will help, of course, to standardize and simplify the analysis of the data, because as you can imagine, we are talking of 17 SA with many endpoints measured, so it's not so easy. Here is my last slide, and is what we at EFSA level are currently doing. There are other efforts ongoing, in particular on transferability of the site from this academic lab to CROs or even GMP labs. Testing, there is a lot of testing ongoing, as you will see in the next slide, and to conduct more IATA or more case studies for more complex scientific situations. So from the EFSA side, we are testing, but I have to say that we are testing only pesticides and only pesticides approved in Europe. We are producing transferability data. We are facing also some issues with the test systems accessibility, but we are having a project that we hope will solve this. We are also working on interpretative guidance for our context of use, that is the pesticide risk assessment, in particular for quantitative methods. And there are other research projects ongoing in terms of developing AOPs or further molecular characterization of the test system or of some of them. So this together with the other work that is ongoing is how EFSA plans to contribute to these efforts. So just in 4 sentences, as you have seen, as I have presented, at EFSA, the DNT in vitro battery has been successfully used, has been accepted and trusted by the different stakeholders, from industry to our risk managers. And it has provided relevant and high quality data for assessing DNT potential of chemicals and pesticides in particular. And it is clear that can measure early cellular processes that are difficult or cannot be measured with the current in vivo models. In the pesticides risk assessment in Europe, we have the obligation of integrating all the public available evidence, so the DNT in vitro battery will be integrated. For further regulatory action, the transferability from to naive laboratories and able to conduct the sales under GLP and make them commercially available is needed. As you have seen, the AOP for IATA is our current recommended framework. And the final aim of the OECD work is to have a further interpretative guidance, but also an agreed standard workflow for the weight of evidence. And you will see how we are working on this. And just finally, but not least, I have to say that this will continue to imply the international and the collaborative effort between the sites, developers, and also the regulators. And at least for EFSA, this is paving the way in a way for the broader use of NAMs or in vitro methods in general. I thank you very much for your attention.
Thank you so much. And this time we'll open it up to clarifying questions. Again, if we can use the raise hand feature, that's helpful. Okay, Dr. Sura.
Thank you, Dr. Iris, for the wonderful presentation and an overview of everything that you shared. Just a quick question. Can you elaborate a bit on the cell types that were part of the in vitro battery? Are they sourced from humans, rodents, or?
Thank you very much for your question. So in a very broad term, I think my colleagues that will present later will give you more details, but we have most of them human, but not all of them human. There are some rodents.
Okay, it doesn't look like we have any more clarifying questions at this time. If we can, again, how the Q&A feature does not work. How we indicate that we want to have a question is we use the raise hand feature. Okay, so we're going to move on to the next speaker. Again, all of the Cicatrum discussion will be at the end of the session. I would like to welcome Dr. Helena Holberg, and she's going to give her talk on integrated approaches to testing and assessment IATA case studies for DNT.
Okay, thank you. Thank you, Iris, for setting me up for talking about the IATA case study. And I'll see if the IATA case study that we at DNT has been working on, DGT. First, before I'm starting, I just want to acknowledge everyone that has worked on this, the NICEDAM team, of course, and then also the DNT ATI team, which you're going to hear a little bit more about from Chris McPherson later. But I also want to acknowledge the DNT in vitro battery contractors. So you already heard from Iris about the guidance document or the initial recommendation, the guidance document, which is informing on the DNT in vitro battery and how to use it and the interpretation. So there is also information about the DNT cell types, for example, very, very nicely described there. And I could answer that question if you want later. We can talk about that. But also in appendix to the guidance document is IATA case studies, which is more exemplifying different regulatory needs. And the one I'm going to talk about is the prioritization that was established by DGT or developed by DGT. This was published in 2023, but since then we have actually expanded on this one because we found more information in the literature and also based on some of the feedback from the reviewers of the case study. So we just recently published a paper with the same name, so DNT to prioritize aromatic organophosphorus flame retardants. So I'm going to guide you through this and show you how this kind of approach can be done. So this is, again, showing the key processes of neurodevelopment that are covered in the DNT in vitro battery, that OECD guidance document that Iris also talked about. So we have proliferation, migration, differentiation, nevered outgrowth, synaptogenesis, and then neural network formation and function, which if these are happening in the right way and not being perturbed, you should also have a functional brain. And of course, what we're looking at the endpoints in vivo is mainly behavior and also in humans. So these are the DNT in vitro battery at the OECD. I don't have the synaptogenesis here because we did not have data for that in this specific case study. We could add it on later. But we also have some additional assays that we have incorporated. We have nevered outgrowth, 2 assays from NCATs, and we also have neural network acute activity, which is not really a DNT effect, it's more an acute effect, but we also included this because we thought it could give some additional information. And then we have some small model organism behavior assays. We're looking at planarian behavior as well as zebrafish behavior assays. So this is the framework for our IATA case study for prioritization. Our problem formulation was to select high priority compounds within a class of organophosphorus flame retardants for further DNT hazard evaluation. So first gathering all existing information, and we here used the literature and ICE. And then because we do not have a lot of information from those studies, we do kind of a weight of evidence assessment and then If there would have been enough, we could have stopped there, but there was not enough, and we then generated screening data information using the DNT in vitro battery and the small model organism. And then again, see if we have enough information, but in our case, then we wanted to use this kind of screening information to decide on what tests to do next if we didn't have enough evidence. And in this case, we could use mechanistic, or maybe in my mind, preferable mechanistic in vitro assays, but we could also do tailored novel in vivo assays, or if needed, a traditional DNT in vivo study. So what data were we using for this? So here are just a little scheme showing the workflow collection of data. We use the OECD DNT in vitro battery, as I mentioned, and therefore there we have our own pipeline where how we analyze the data, so we get benchmark concentration. I think Chris will mention a little bit about that. But then we had our DTT DNT battery, which was, as I mentioned, a few additional assays as well as some small model organisms. Again, we're using the same pipeline, the benchmark concentration pipeline. From the literature search, we took data both in vitro, small model organisms, and in vivo data. And here we had to just trust what the authors had been provided. So we tried to use the lowest observed effect concentration to be as close to benchmark concentration as possible. And of course, there is the higher uncertainty because we don't have normally the raw data. And in this case, we did not do a bias analysis because this is for screening and prioritization. But if you want to use it for hazard, you would probably do a bias analysis, which we're doing in our follow-up IATAs. But then we also used ICE. And you know about ICE, and you will have a whole session or at least a talk about ICE, which is having curated high throughput screening data. And we also used that information, and there we use the ACC. And I know Sue asked about this kind of things previous, like what kind of are you using AC50 or using ACC and benchmark concentration? We decided to use ACC because it's most similar to benchmark concentration, and we wanted to compare and integrate this data. And actually, some of the data were looked for the same assay that are ACCs from the Toxicological pipeline. And there's actually no difference really between BMC and ACC in the cases where we had the same pipelines looked at. And these are for these compounds. Of course, other compounds might be different. But then we also had a literature search where we looked at exposure and biomonitoring data as a human data, where we then used PBPK modeling to derive a Cmax concentration in the plasma. So which were the chemicals we looked at? We looked at novel organophosphorus flame retardants. You can see them in this table here. If this works, yes. But online they won't see it, right? Oh, they do. Perfect. Okay. So these white ones here are the aromatic organophosphorus flame retardants that we wanted to prioritize. But we wanted to anchor them, compare them to some chemicals where we had a little bit more data, and therefore we used the brominated flame retardants as well as the aliphatic halogenated flame retardants that you can see on the top there. So why did we look at this class of compounds, and why did we compare them to these other ones? Well, the brominated flame retardants have been phased out because we know that they are developmental neurotoxic or neurotoxic. There are also other health concerns with those compounds, so they are not on the market anymore. Instead, they have been replaced with organophosphorus aromatic flame retardants. But so there's a continual rise in use and therefore an increased human exposure of these compounds. But we have very little information on the toxicity of these compounds. But there is a concern because they resemble the structure of organophosphorus pesticides, where we know several of them have DNT and neurotoxic potential. So why we did a prioritization of this and why didn't we just study them all is because there's a class of compounds, 20 to 50 compounds. They are also available as commercially as asymmetric mixtures. So it would be really impossible to test all of these combinations and all these compounds in an in vivo study. And we also know that in vivo study is actually enough sensitive or good enough for picking up some of these effects. So therefore, we wanted to do a screening approach to prioritize chemicals for further information. So I'm not going to talk about ICE because you can hear about ICE. I just want to tell you what we were doing with ICE, and you all know ICE. It's amazing. It has a lot of high curated data and a lot of high throughput screening data. What we did, we used the curve surfer and we searched for all the assays. We didn't only search for DNT. You can in ICE search for specific endpoints, but we didn't search only for DART or just DNT. We searched for all because we wanted to see if we could find some additional mechanistic information and try to understand if other endpoints were more sensitive. So we used curve surfer and did that. And then we analyzed, well, we looked at all the data, but we analyzed specifically the ones that were more sensitive than the DNT in vitro battery. So what does the data look like then? So this is just a summary of the findings from the DNT battery. What you can see here are the different organophosphor or the different flame retardants. In the lower here, you can see the different endpoints, and we have now grouped them based on their key process they are modeling. We have proliferation, differentiation, differentiation, network outgrowth, electrical activity or function, and then behavior assays. And what you see here is a lot of colors, which is the benchmark concentration. So it's the concentration which give an effect. It goes from the lowest yellow up to the dark purple. You want to look at the ones in the middle here. These are the organophosphorus flame retardants that we wanted to prioritize. And what you can see is a lot of colors. So overall, as a class, they were quite active in the DNT in vitro battery. And if we then compare them to the brominated flame retardants, which are the ones on top there, you can see that they're quite similar. They have similar colors and similar numbers of acids are active. But of course, there are sources of uncertainty and we need to identify those. And I'm going to just highlight a few of them, not all of them. The first thing here in this, I didn't mention it, but the white is inactive and the gray means it's not tested. So in this case, for this compound, we actually had not tested. It was not tested in all the assays. So of course, it's an uncertainty. We don't have the data. There's also a lack of concordance between assays that are looking at the same key process. And two examples here are network outgrowth. You can see we have a lot of assays measuring the same key process, and some of them are white, some of them are colored, which means we don't have a concordance. However, these are, it is mentioned this as well, there are different, these cell systems are different, they're different species, they are different cell types considered in here. So some of them are simple cell lines, while some of them are primary cells, some of them have other cell types present. So this is most likely a biological difference, since it's not something that we maybe need to be so concerned about. We need to know a little bit more about it, I would say. Some are in 2D, some are in 3D, and they are also looking at different windows of development. So these things could be more studied, and there are ongoing work from the methods developer to look at what pathways are actually perturbed or can be measured in these systems. But the same for migration. Migration, I think, is much easier to really say with the biological differences, because we're looking at different cell types. They will be perturbed differently because they have different reasons to migrate. But other things are, Sue also mentioned this the other day. Here you have a compound where we will have one hit in one assay. What do we do with this? This is also, of course, an uncertainty. In this case, I would say we wouldn't probably prioritize this in our case because we were interested in testing the ones of most concern. But depending if you are a company, you might want to could test this one further because you think it's more safe than the others, and you might be so it's up to the person that are going to use this data. But then we also, and it is also mentioned, is we have some mechanistic uncertainties. Not only do we lack some of, or we're not measuring at the moment some of the key processes that are important, myelination, for example, different receptors. We can measure some of those things in here. We just haven't developed the test assays for it, and there's ongoing work in doing that. But one other thing is that these assays are actually looking, if you're thinking about an AOP, it doesn't necessarily looking at the mode of action and the MIE. It's just looking at later key events. Like we don't really have early key events either. So by integrating additional information, we hope to lower this uncertainty. And that's what we try to do with the ICE. So I'm just giving one example. We did this for all the compounds. Here you can see on the y-axis the point of departure in micromolar, and then you can see we bundle them together based on the key process. So the first ones are from the DNT in vitro battery, proliferation, migration, differentiation, and with outgrowth, network function, and behavior. But then we also incorporated ice, and we only took the ones that had lower concentration here. Otherwise, we would have a very high bar, or the x-axis will be really, y-axis will be really long. And then we also incorporated the literature data. So what you could see here, just looking at the DNT in vitro batteries, that we see in the zebrafish is actually the most sensitive in this case for this class of compounds. So when we incorporated ice and literature data, we actually, this line is showing the lowest point of departure from the DNT in vitro battery. You can see that we had some assays that had lower point of departure from ice and the literature. And we looked at those, what those endpoints were. They were mainly annotated to glial differentiation, the immune processes, and the endocrine system, which are things that some of them you can actually measure in the DNT in vitro battery, but we are not focusing on measuring them right now. So again, I mentioned this already, there's some higher uncertainty with the point of departure from the literature data, so we would maybe not incorporate too much of that. But integrating of this data actually increased our confidence and can also provide a more mechanistic understanding. Final here, looking at the relevance to human exposure, we look at biomonitoring data from where we had the plasma, which was only for 2 of the brominated flame retardants, where we looked at data from breast milk, child hand wipes, and house dust, as well as urine, and then we used reverse PK modeling to see what the concentration in the plasma would be from those exposures. And those are the colored bars that you can see. Again, these are the organophosphorus aromatic flame retardants that we want to prioritize. What you can see here are in the triangle, it's the DNT in vitro battery, the most sensitive endpoints. You can see they're quite similar for all of the flame retardants, so the older compared to the aromatic flame retardants. We had, for some of these compounds, we have point of departure from in vivo, and we plotted them here just to compare. And what you can see here, also those are in the circles here, that they are an order of magnitude or actually even overlapping sometimes with the DNT in vitro battery. Most of the time when there was a difference in DNT in vitro battery, it was more sensitive. And then looking at the human exposure, I will say that one uncertainty here is that we don't have a lot of human exposures, but so there might be even higher concern of this. But when we have the data for some of these compounds actually overlapping, the in vitro point of departure is overlapping and in vivo with the human exposure, which could be considered a concern for these compounds. What DTT did, and this was actually before I started, they prioritized 2 of these compounds for tailored in vivo studies, IPP and TPHP, based on the in vitro battery data, because the other data we actually incorporated afterwards, but also based on the structure of these compounds, because in the end, they could have picked any of them because they were quite similar, but TPHP is one of the ones that we are most concerned of, especially looking at also the exposure that is quite high. So a quick summary, organophosphorus flame retardants, the aromatic novel substitutes, they have comparable in vitro activity to the older flame retardants. The in vitro activity was within an order of magnitude of in vivo. Point of departure is usually more sensitive when we have that data. Activity concentration in the IATA overlap with predicted human exposures for some of these compounds, which could be a potential concern. And when we incorporated ice in the literature, we identified other sensitive targets, such as endocrine disruption and the inclusion of astrocytes and microglia cell populations, which might be where we want to do additional cell assays that could be incorporated in the battery or done as a second tier in this case. We lower the uncertainty with integrating data from multiple sources and also provide more mechanistic understanding. And in this case, IATA suggested compounds such as TPHP to be prioritized for further testing this is really the final slide, which is just talking about the next steps, which I think is going to be really important. The next steps to expand the regulatory applicability of the DNT. So there is a project at the OECD, which is the DNT IATA framework template and guidance of support, which EFSA and NICETAM are leading this project. It was just approved. And the aim of this project is to develop an IATA framework template specific for DNT. And in this way, we're going to advance and provide guidance first on addressing quantitative in vitro to in vivo extrapolation, which is maybe not everybody has experience with, but give some guidance on how that can be done. We also want to work at standardizing uncertainty analysis for integration in the weight of evidence assessment. So we're going to use the IATA, so what EDIS have presented, for example, as well as this prioritization, but there are also other IATAs, and try to discuss on how we can come up with a more streamlined approach. It will be easier for others to develop new, and also it will be closer to a defined approach because it will always be done in the same way. So again, we're going to have then the things we are going to consider here is several information sources, multiple problem formulation, and have a consistent way of integrating data depending on the context of use, of course. And there is a workshop that is taking place in October, so quite soon, where we are going to have breakout groups also to discuss this. I think it's going to be very, very interesting how we move forward with this, and we're expecting to finalize it and have it approved by the end of 2025. We'll see about that. But so thank you. Happy to take clarifying questions.
Thank you. Please use the raise hand feature to indicate that you would like to ask a clarifying question. Dr. Kleinschreier, go ahead.
This just occurred to me. Do we have any plans to try and expand the exposure collection data for some of the other flame retardants? Because it had quite a few sources for something like PPHP. There was the breast milk and the house dust and the cord plasma. Are there any efforts to actually expand out the exposure data for some of the others?
No, but that's a good point. We can do. I mean, we have talked with Heather Stapleton, for example, and have been getting all her latest data, but I'm sure there are more data out there. You might be aware of some data, for example. So yes, it's a good idea, especially for the ones where we have very limited information. It would be good to incorporate as well.
Thank you, Helena. A quick question. You mentioned about, can you hear me okay? Closer? A little bit, like you mentioned about the lack of concordance, but have you looked at, say, oligodendrocytes? Like you have assays for capturing the migration, differentiation, and then the myelination. Given that they are the cells which are responsible for myelination, is there any correlation or concordance between what you're seeing for these developmental processes?
So for the oligodendrocytes, we do have migration and differentiation. We do not have myelination because it's happening later. I have developed a model myself for myelination, and there is discussions of incorporating the people that are continue to develop that assay as well, because it's important, but it's happening later in stage. There are some concordance with, but not for all chemicals. So for the organophosphorus flame retardant, the oligodendrocytes were actually more sensitive than the others, and we think that is due to the mechanism, it's likely endocrine thyroid. But for some of the other compounds that Chris is going to mention, there is actually not a concordance between migration and differentiation of oligodendrocytes. So it all depends. And that assay, the only assay that have oligodendrocytes is the neurosphere assay. So they have they also have neurons and radioglia incorporated. So they are also, they are communicating with each other as well, right? So that it's a more complex system, which I think is both good and a challenge sometimes.
Thank you so much. I'm not seeing any more clarifying questions at this time, so we're going to move ahead with the next speaker, who is Dr. Schaefer, and that presentation will be remote.
and he will talk about the DNT in vitro battery.
Okay, well, thank you for the introduction and good morning, everyone. I'm Tim Schaefer at the U.S. Environmental Protection Agency. I am going to talk a little bit more about the battery and focus a little bit on establishing confidence in the data that come from the battery and give some brief additional case studies. So here's the outline. A quick introduction to the battery. The previous speakers have already covered it to a certain extent, so I won't dwell on it. But I want to spend the majority of the time talking about how the battery aligns with the different criterion for confidence in the use of data from NAMs. And as I said at the end, present a couple of brief case studies and then a conclusion. So as was mentioned previously, the document, the initial guidance was approved in October of 2023. And this was important because it recognized this battery of in vitro assays for developmental neurotoxicity. It provides some international recognition and credibility to those assays, as there are 38 different member countries of OECD. And so this is kind of the first step in moving towards a formalized approach. And so this was a event that, as Iris showed, had been worked at for a number of years prior. This is a real quick overview of the battery. There are assays in the battery. They come primarily from the University of Constance, from the EPA, and from the IUF in Dusseldorf, Germany. The vast majority of these assays look at structural endpoints using different cellular models in vitro. Most of these are human cell models, as was mentioned before, and I'll talk a little bit more about this. There are a couple of rodent models that we do use. Most of these assays provide structural information, but there is one assay, the rat network formation assay, that does provide information about the function of networks as they develop, and I'll talk more about that. But now that we have these assays and this battery of assays, how do we begin to establish confidence in those? We really need a roadmap to do that. And part of that involves, I think, having case studies that demonstrate the utility of the battery. So Amy Clippinger mentioned these two different paradigms for establishing confidence in data from in vitro new approach methods. They're very similar. It's kind of like Google Maps versus Waze here. They have a number of the same criteria. And so what I'd like to do is I'm going to use the one by Van der Zalm and kind of walk through the DNT in vitro battery and show how it aligns with these different criterion for establishing confidence. I do want to briefly remind everyone that the underlying premise of these assays is that they evaluate effects of chemicals on processes that are critical to development of the nervous system. And Helena presented that slide. So those processes have to take place throughout the nervous system at different times in different places in order for it to develop properly. And if they're disrupted, then that provides a possibility for neurodevelopmental outcomes. So let's talk about why these assays were included in the battery, because that already begins to address some of these confidence criteria. So each assay had to be deemed ready for use in screening and prioritization in these reviews that were conducted by Fritchie and Ballprice and Sachana. And they had to, in order to be included in the initial guidance document, test a common set of chemicals across all the assays. That data then had to be provided to the US EPA so it could be put through the Toxicological pipeline, which is a defined approach for analyzing data. And I don't mean that in the OECD defined approach terminology. I mean, the pipeline is a transparent approach for analyzing data. And all the assay developers agreed to provide these very detailed methodological descriptions, sorry about that, about the assays. And these are called tox temps, and I'll present a little bit more information about those. But already here, the fitness of purpose, independent review, data transparency and integrity and technical characterization are starting to be addressed just by what was required for the assays to be included in the battery. I want to talk a little bit more about independent review. So all of the assays that are in the battery have been published in the peer-reviewed literature as a primary publication describing that assay, describing the biology of the assay, the performance of the assay with small sets of chemicals. And those are published in peer-reviewed journals. For example, these two publications come from Toxi, as does this one by Harrell. So there's independent review in the peer-reviewed literature. And then this paper by Ball Price was a group of scientists who evaluated the different assays based on these 4 categories, developed a ranking system, and then gave the assays grades for how well the method was developed, how well it performed and how replicable it was, and whether or not it was ready for screening. And the assays outlined in green here are the ones that are included in the battery. And so again, this was another level of independent review, although I will note that there were a couple of the assay developers among the authors of this paper, but a lot of the authors and the people that did the ranking were not people that developed the battery. So there's also a pretty extensive review of the battery as a whole. And so this is the timeline for when the OECD WNT accepted inclusion of the DNT project in its work plan. The draft guidance was developed over a 2 or 3 year period. It was then reviewed by expert group comments. A second draft was produced, and that underwent 3 rounds of comments and revisions by members of the WNT. So these are the member states of OECD. These include organizations like ECHA, like EFSA, the Office of Pesticides and Office of Toxics at EPA, Health Canada, as well as others. So this was an international and relatively comprehensive review of the battery that was conducted. At the same time, the EPA in 2020 conducted a peer-reviewed or a scientific advisory panel that focused on the assays of the battery. And then there was this publication that came out in 2023. This was a opinion effort, I believe, where they had an expert panel that was convened that reviewed the battery. And so I'm not going to read all of these things, but I want to just highlight some things. These are comments from all 3 of the different reviews that I mentioned. It mentions using it as a screening tool. You want to employ as much data as possible to inform DNT potential. Integration of data across different methodologies, in vitro, in vivo, epidemiological, clinical studies is important to detect the accuracy and sensitivity of DNT. The initial guidance states that the battery should be used for screening, prioritization, and weight of evidence. And in the EPA SAP, that panel concluded that whether the data is screening or mechanistic, data from in vitro assays, as long as they're published in peer-reviewed publications, can be used as weight of evidence. And so I think there's consensus that the fit for purpose of these assays is screening and prioritization and weight of evidence decision making. And so again, that addresses these 2 components of the framework. And all 3 of these reviews indicated that the battery should be a living process that evolves as the science evolves and moves forward. So let's talk more about data transparency and integrity. And this is just a quick summary of all the different sets of compounds that are being tested or have been tested in the EPA and the DNT-IVB assays. Initial testing focused on testing DNT reference compounds as well as negative compounds, chemicals with in vivo DNT guideline studies, as well as chemicals of programmatic interest. And you can see where the data are in terms of how available they are. And so as these chemical sets complete their testing, most of them, if not all of them, are being made available through the ToxCast dashboard. And so this figure is one that's been published many times. It shows the lack of data for most environmental chemicals that are shown here on the left, where we have very few of these that have been tested in DNT guideline studies. In fact, there are about 220 that have been tested in guideline studies. Whereas right now, they're currently available in ToxCast data for 81 chemicals in the battery. From this phase 1 set of compounds that the Division of Translational Toxicity at NIHS sponsored testing for, there are about 115 more that should be available soon. And then there's currently about 105 that are undergoing testing. Data from those will be available in ToxCast in 2025. And for some assays, my slides will catch up with me here, we actually have data for over 400 chemicals that are available in ToxCast. And so if you compare this to the underlying data for guideline DNT studies, most of that data is not available for public review, or if it is, it's very difficult to get. And so this provides a level of transparency to the data that are there for the DNT in vitro battery that's just not available for guideline studies. This is a summary of the ToxTemp form that I mentioned, and this gets to the aspect of technical characterization. And so I've highlighted here because this ToxTemp form is a quite detailed summary of the battery, or of each assay in the battery, and the different aspects of those that speak to characterization of the technical capabilities of the assay in the battery. I would also note that within ToxCast now, they have developed OECD 211 forms for all of the assays that are in ToxCast, which includes the DNT in vitro battery assays. And that information will be available soon online as well. And so that's additional technical characterization. And among things that are found in here are things like the coefficients of variation, standard deviations of control wells, Z prime scores for some of the assays, and so it tells you how well these assays perform. So I want to then focus on biological relevance here in the last couple minutes. And specifically, the guidance or the framework says human biological relevance. But if you go into this, it says demonstrate the similarities between the physiology of the test system or the biology measured by the test system and human biology. So somebody asked earlier about which assays were in rodent cells versus human cells. Most of the assays that are in the battery are in human cells. There are 3 assays: the rat neurite outgrowth assay, the rat synaptogenesis assay, and the network formation assay that use rodent cells. Before I get into those, I want to step back to the relevance based on the key neurodevelopmental process. So this table shows those processes on the left-hand side. On the right-hand side, it shows different types of clinical conditions that the disruption of these neurodevelopmental processes can contribute to. In the middle, there are examples of different types of chemicals or insults that cause disruptions in these processes. Now, I want to be clear, I'm not saying that any of the chemicals in this middle column cause these clinical outcomes, but that they are known to disrupt these processes. And when you disrupt these processes, that disruption is known to contribute to these different clinical outcomes. And so that provides a level of human relevance. Also, you can take the different key neurodevelopmental processes and align those to common measurements that you might examine in a in vivo study. Changes in neuropathology that are caused by differentiation changes, different types of histopathological changes could result from these disruption of these different processes, and so on and so forth. So the processes that underlie the battery do have important and relevant links to outcomes in either clinical conditions or outcomes that you might measure in an in vivo study. Let's focus in a little bit more then on these three assays that are using rat primary models. And I want to take a few minutes and talk about using the network formation assay, how the processes that are measured by this assay are relevant to human neurodevelopmental biology, because that's really the criterion that we need to meet, is that showing that it's relevant to human neurodevelopmental biology. So for those of you that aren't familiar with the network formation assay, and I'm going to use this as an example because the neurite outgrowth and synaptogenesis assays use the same model, and those processes contribute to network development. This uses a specialized plate that has microelectrodes in the center of each well. There are 16 of these. You can plate electrically active cells on top of these electrodes and then measure their electrical activity, and that's what's shown here as a snapshot of that activity. So these are extracellular action potentials. Each box here in the snapshot represents the activity on one electrode. And the way the assay is done is we plate primary cells on day in vitro 0. We expose them to toxicant 2 hours after plating, and that toxicant is present throughout. We make recordings on day in vitro 5, 7, 9, and 12. And we change the media and because we need to feed the cells and renew the chemical on days 5, and 9. At the end of the recordings on day 12, we measure the viability of the cells that are present in each well. And so just as a first comment towards the relevancy of this type of measurement towards human biology is what we're actually measuring here are the underpinnings of EEG recordings. So these types of electrical action potentials are being summed when you make an EEG recording. And so the biology that's driving these changes in the MEA and the EEG is identical. It's action potentials and the summation of action potentials across time and space in the EEG. So in terms of what types of neurons are present in the cortex here, the primary cortical cultures, we have both neurons and astrocytes. We have excitatory and inhibitory neurons. We see synaptic terminals that are excitatory and inhibitory. You can actually stain those synaptic terminals. We do see the presence of sparse microglia. We also know that different receptor pharmacology is present by measuring the functional responses. And we do know that there is expression of some P450s that if you look at what's in the cortical culture versus what's in the frontal cortex, you can see that they're very similar and they're very different from liver on the same times in vivo. And so the biology here is very similar to the human biology in the cortex of the brain. Well, development of network function is critical for neurodevelopment across species. And the spiking, bursting, and intrinsic activity or activity that develops are intrinsic to development of the network. And this happens spontaneously both in vivo and in vitro. And we know that neurodevelopmental processes are influenced tremendously by electrical activity and vice versa. And that these the synchronous activity in networks is is integral to sensory awareness, attention, memory, and other cognitive processes. And we know that these patterns are highly conserved. And so there's actually literature that shows that there's greater similarity across brain regions of different species than between different brain regions within the same species in terms of the electrical activity. And that's shown here. So these are recordings from MEAs from a human model, from the rat primary cortical neurons, from the midbrain and spinal cord of rats. And you can see the difference in the patterns of activity. And you can see that the human and rat cortical are much more similar than the midbrains. And so just a brief explanation of what these recordings are. These are what are called raster plots. Each horizontal line is the activity on one electrode in a well. And every time you detect an event, you make a vertical tick mark. And so it gives you that pattern of activity over time. And you can see how different these patterns are, and that the cortical pattern in primary neurons is from rat is very similar to the pattern from human cortical model. So in terms of case studies, Helena just presented right prior to me, this case study on the flame retardants. We've also used assays from the EPA part of the battery to characterize 150 different PFAS compounds and prioritize them for which ones may potentially alter neurodevelopment. So those are two examples of screening and prioritization. And then in terms of weight of evidence, there are several examples now of assays from the battery being used for weight of evidence. There was a guideline waiver study for glufosinate isomers that's published in Dobroniecki et al. In 2022. Iris talked about the deltamethrin and flufenicet case studies. Dichloran, which is a little bit more of a focused effort, they decided to require a guideline DNT study based on weight of evidence, which included positive effects in an assay that's not part of the battery, but was mentioned earlier. This is the acute MEA data showed effects of dichloran, and that was used as part of the weight of evidence to require a guideline study. So the in vitro assays can be used to both support in a weight of evidence manner a waiver or to require the guideline study. And then recently, the EPA has been doing the reevaluation of organophosphate neurotoxicity. As part of that, we've been used data from the battery to evaluate the DNT potential and inform the relative sensitivity to ACH inhibition to inform FQPA determinations, and then do individual organophosphate weight of assessments. And so we've already released, I think, acephate and methimidofos, and that there will be others coming out in the near future. And so there are already a number of different examples of the case studies for using the data from the battery. I want to take one quick example and show how it can have a tremendous impact. And so this is the glufosinate example where we evaluated whether or not a guideline study could be waived for isomers of glufosinate, which is a common herbicide. So this timeline shows when the Office of Pesticides requested that ORD collect data from the battery to the time that they made a decision to waive the guideline study. And that was just about 2 years, a little bit over that. If they had requested guideline studies at the same time they requested ORD to collect data, it would have taken at least 3 years, maybe more, to get the data in and be able to make the decisions on whether or not to, what they would do in terms of the glufosinate risk assessment. In terms of the animals used for the in vitro studies, we used 3 pregnant dams and about 12 to 15 pups. 2 guideline studies, because there were 2 isomers of glufosinate that were being considered, would have used 160 pregnant dams and over 1,600 pups. And in terms of cost, it cost ORD about $97,000 roughly to do the in vitro studies, whereas 2 guideline studies probably would have topped out over $2 million. And so you can see the tremendous advantage of having the data from the in vitro assays to helping to make the weight of evidence decisions around glufosinate and the waiver. So I hope I've convinced you that the DNT in vitro battery meets many of the criteria for establishing confidence and use of that data. There is still work to be done. Megan is going to talk in a few minutes and talk about work ongoing to transfer assays from one laboratory to another. I think the review process for the battery has shown that there's consensus that it can be used, data can be used for screening and prioritization and weight of evidence. And I hope that the case studies, help to document the use of the battery in these contexts, and that the case studies can help speed decision-making, reduce cost, and contribute to health protective decisions. There's consensus behind the fact that the science for the battery will continue to evolve and improve, but that does not need to delay implementation of the battery at the present point in time. And so I just want to recognize that there were a lot of people who have contributed to this work, and they're shown here, and I would be happy to take any questions.
Thank you. And we will open it up for ICFAM and SICATM members to ask clarifying questions if there are any. And again, please use the raise hand feature. I do not think that there are any clarifying questions at this time. So with that, we will move on. Oh. Okay, so we'll introduce Dr. McPherson, who is going to provide a talk about integrating screening level D&T information of chemicals in a NAMS battery to identify compounds for future study.
Okay. Thank you for the opportunity to talk today. This is my first CICATM meeting, so it's pretty exciting, learning a lot. Today I'd like to talk a bit about how we're using data from NAMS to identify compounds that are with potential D&T hazard. And I would just like to first acknowledge my team here. This is a pretty large group of very talented individuals at NIEHS that have been working on our D&T project. And this team is the known as the Developmental Neurotoxicity Health Effects Innovation. This team was established in 2019, and we're formed around kind of four main program objectives. The first is to generate screening level information using NAMS as an interim means to evaluate hazard and prioritize chemicals for further evaluation. We want to incorporate human relevant mechanistic, behavioral, and brain network assessments to hopefully address complex neurodevelopmental issues. We want to hopefully contextualize our in vitro and in vivo findings with human exposure using IV, and other in silico approaches. And the biggie is just to establish communication pipeline with our numerous stakeholders, with published manuscripts and as well as sharing our data publicly to make our data available with the concerted effort to help the global progress of the field of developmental neurotoxicity. So as the other people have mentioned, the other talks today have mentioned as this D&T battery. So this is the battery that we're using. Tim and Helena and Iris gave a great detail of this. Well, anyway, so this assay that we're using. So again, just to not get into it too much, but we are using the human-based 2D and 3D models as well as rodent primary cells. And our battery differs a bit from what was previously described, and we have included a zebrafish assay of neurobehavior. So using this battery, we reached out to stakeholders to nominate. So the main objectives of our screening efforts were to screen chemicals for D&T potential in a battery of assays that covers the key neurodevelopmental process. We want to evaluate these assays for redundancy. We want to develop ranking methods to evaluate and compare chemicals for the degree of D&T potential. We want to come up with ways that we can hopefully prioritize chemicals for further testing and other targeted either in vitro mechanistic studies or more hypothesis-driven in vivo studies if necessary. And again, integrate the data into our public sharing platforms. One is the D&T Diver, which is an online tool to visualize our our screening information and to, again, share with our global stakeholders. So we sent out a call to individuals within the DNT community to nominate chemical libraries for testing in the battery. And we received a large number of responses. We got over 700 compounds that were nominated, and we, as the team, selected these compounds using different selection criteria, including whether they had evidence of DNT in vivo, if there's known human exposure, if there were guideline studies complete and then lacking in vitro data, if there was incomplete in vitro battery data from these compounds, and if the compound were suggested by multiple stakeholders. So as I said, we got a number of compounds that were nominated, and we broke them into groups. So we have 3 phases of compounds. We have phase 1 and phase 2, and we were just approved for a 3rd phase of chemicals. So what I'm going to show today is data from our first phase of chemicals, 115 chemicals that were run through the battery. They include a diverse range of chemical use classes, including insect repellents, PFAS, flame retardants, which Helena talked about earlier, insecticides, herbicides, fungicides, some drugs, chemotherapeutics, as well as HIV therapeutics, some botanical compounds, and then we also included some compounds that are suspected to be DNT negatives in vivo. That's a little harder to find sometimes. And then also included some compounds that have evidence of being DNT positive in vivo. So yeah, that's the first phase. And I just wanted to mention that we do have a second phase of chemicals that are being run through the battery right now. And then that's, they're about, I believe, 70% complete. So these are being run through the various laboratories. And again, we have another diverse group of chemical use classes in this set. And we then also tried to include a lot more of what we thought are DNT negative compounds in this group. So this is an example of our data analysis pipeline. So the substances were screened in multiple concentrations in all assays. We received the data, it went through this various QC checks, loaded it into our database, and then it was sent to our data analysis people who did all the heavy lifting. And I want to give a shout out to Ray Wasse and Skylar Marble, because they did all this amazing work, that the data that you're going to see today. And they were responsible for doing the BMC calculations, doing selectivity calculations, an activity summary of the compounds within the assay. And we use this data, again, as I mentioned, to try and use a couple different methods to prioritize the compounds for DNT potential for further studies at the DTT. And those included ToxPi, the Toxicological Prioritization Index, and we used a multi-objective optimization tool called the Pareto Front optimization. So just to give an idea of what the BMC data looks like, this is a heat map, very busy. The take home that I want you to take home is that we observed a lot of activity in all chemical classes and assays in the battery. And see if I can get my pointer to work. Oh, there we go. So this table is arranged along the top here. You have the various battery endpoints, including proliferation, apoptosis, neurite outgrowth, migration, the MEA assay, and then the zebrafish assay here. And along the left column here, you have the chemical use categories. And right here, each assay has its own cytotoxicity endpoint. So that is used to see obviously how toxic the chemical is to the cells of interest, and we use that cytotoxicity data to help determine whether the assay is selective for the endpoint of the key neurodevelopmental process, or if it's causing overt cell death, and the effect we see is due to just general toxicity. So as I mentioned, so then when we take it that step further and look at selectivity, this is another heat map that shows the selectivity of the chemical compounds across the various assays and chemical use groups. And again, like the BMC data, we saw a lot of selectively active compounds across many of the chemical use classes and the batteries. We did see the most selective hits in assays of neurite outgrowth and oligodendrocyte differentiation in this battery. And if we just further break that down, I just took a summary of the selective hits here by compound class. And so what you see here is on the top of the graph are the compounds. So we had drug, fungicides, insecticides, and herbicides had the most selectively active hits in the assays. And that's shown by the red bars in these graphs. So red means selectively active, and again, that means that we were seeing effect in the neurodevelopmental endpoint before we saw an effect in the cytotoxicity range. And then the yellow bars represent non-selective active. So that means we may have seen activity in both the neurodevelopmental endpoints as well as the cytotoxic endpoints. So there were some assays that had a lot more overall general activity than the others. So for instance, the MEA-TIMS network formation assay showed the most activity, both selective and non-selective, with this particular group of compounds. So from this data, we then, as I mentioned, we used some other different modeling approaches to try and make the list a little more user-friendly for the hits. And so we tried different ways to prioritize, see if we could get a prioritized ranking of the compounds using the ToxPi, which again is the Toxicological Prioritization Index. And if you're not familiar with these, we basically use modeling to generate a way to visualize the activity of these compounds within the assay. And we generated ToxPi models based both on the BMC potency values as well as the selectivity ratios. So just to orient you with this, when you're looking at the pies, each slice represents a specific endpoint within the assay battery. The greater the length of the slice from the center axis means it has the greater potential for, or the greatest activity in that assay endpoint. And so potentially the greatest potential for effect in the DNT endpoint. So I pulled out some examples here with some of our compounds with evidence of known to be DNT positive in vivo, rotinote and methyl mercuric chloride. As you can see, those have pies with larger pieces, and then our putative negative compounds, ascorbic acid and saccharin sodium, have these pies with small to no slices represented. So from those numbers from the ToxPi model, we took the means of those, and we plotted the hits here based on selectivity ratio here on the x-axis, and BMC on the BMC on the x-axis here, And again, so you can see that there's the more the higher ranking compounds are going to be higher up in this area here. And from the ToxPi prioritizations, we found that compounds from fungicides, drugs, insecticides were the highest ranking compounds. And then we then used another modeling tool called the Pareto front of ranking method. So this is a multi-objective prioritization approach that uses a few attributes. The mean BMC from the active endpoints, mean selectivity scores, mean activity and confidence scores, and generates a score based on the fraction of active endpoints. So this is a way to prioritize chemicals, again, based on their potency and selectivity, and to give you a graphic, a representation of these Pareto rankings here. Again, I pulled out some of the in vivo positives, rotenone and methyl mercury chloride, and our reported DNT negatives, ascorbic acid and saccharin, just to give you an idea of what a positive hit and a negative would look like. And as you can see, rotenone is a good example here. Around your this bubble plot here, you have the names of the various battery assays, and the size of the circle represents the selectivity of the hit in that assay. So the larger the bubble size, the more selectively active that compound is for that endpoint. And then it's colored based on potency. So the lighter yellowish green color, that's a more potent compound in the assay. So as you can see, rotenone here has a lot of bubbles, large size, and then that green, the green color, so very potent, very selective. In comparison, saccharin, for instance, only has one small hit in one assay, and all the other assays were not active in the assay. And similar to the ToxPi, we rank these as well with a cumulative ranking. And again, the most active, the highest priority compounds were from the drug, fungicide, herbicide, and insecticide class. So we took these high-ranking compounds, and we were very curious to see how selective the DNT assay was, the battery assays were compared to cytotoxicity data within the Tox21 available data. And for 19 of those compounds, we had Tox21 data here. So here on this graph, we have the top compounds that were identified with ToxPi and the Pareto ranking that also had the Tox21 point of departure. So as you can see, the take-home message here is we have the most sensitive DNT battery BMC in the purple box. We have the most sensitive selective BMC in the green circle, and then the triangles represent the Tox21 point of departure for cytotox. And for a majority of the compounds, the battery was more sensitive and selective compared to the Tox21 data. So we're excited to hopefully use data like this to further prioritize compounds for testing at the DTT. And just to summarize, we think the battery is good at covering multiple endpoints, rapid, it's high throughput and reproducible, as was touched on by the talks earlier. We saw activity observed with varying potency across all endpoints and chemical classes. And we think it's well suited for screening and prioritization. And, as has been mentioned before today, the assay, it's an evolving assay, so we know it doesn't represent all cell types necessary for neurodevelopment, but we're excited and we still find it very useful. And in its current form, it's not fit for purpose to elucidate real solid mechanistic understanding, but it can give us an idea of where to hunt for future studies. And again, there's narrow coverage of the chemical universe, and we're doing our best as we put these different phases of compounds through the battery. That hoping we can contribute to the field in that way, as well as continue to evolve studies with the data we have from that within our programmatic interests. And again, I just wanted to outline our team's integrated framework. So the screening battery is a small piece of testing, as Helena touched on earlier. So we're really using the data we get from the screening as a tool to further prioritize compounds for again, more mechanistic studies, so be it more complex in vitro models, or as well as more hypothesis-driven rodent studies, or if necessary, D&T guideline studies. And again, we want to share the data publicly in our two main databases, D&T Diver, SEBS, as well as ToxCast, where Tim mentioned, and within our peer-reviewed publications. And with that,
excellent. Thank you so much. And at this time, we will take clarifying questions from SCATM and ICFAM members. Yeah, Christine.
Hi, Chris. Very nice presentation. I actually had a question regarding your slide where you had the two phases and the different categories of substances that are being evaluated. Could you speak to a little bit more about what exactly botanicals encompass? And then there was a category under phase 2 named mixtures, if you could speak to that.
Yeah, so the botanicals, we tested basically 6 botanicals, I think, with various evidence of neurotoxicity. And I can tell you, I can't get into the specifics right now, but we tested the botanicals that, like yohimbe, kratom, those types that are heavy public use. And for each botanical we also tested at least 2 to 3 different formulations. So we tested formulations of botanicals made from, say, an extract of a root, a liquid extract, or a powdered extract, or say like a whole leaf botanical. And the mixtures were, that comes from a different working group, a complex mixture and exposure group within our division. And there are a number of industrial compounds. Maybe Helena can speak more to the specifics on that. I don't recall, but the lists are public on our website that I can share with you as well. But so we took mixtures. So we made mixtures of I believe it was 3 compounds of each, and then we have the individual compound tested alone in the battery within that chemical set.
Okay, thank you.
Yeah.
I apologize. I did not sign into Zoom, so I did not use the raise hand function as requested. I just have a quick question. If you could maybe elaborate a little bit on the steps you take when you analyze the data. Are you taking the analyses at face value, or are you going in and looking at the individual plots? And I'm asking out of curiosity because my experience, even with just BMD modeling for like acetylcholinesterase, we often have to do the visual fit looks at the data. And so far we're seeing similarly when we're doing like the pipelining, some of these, yes, you can get a number, but then you look at the data and it's trying really hard to fit something. So I'm just curious if that is part of your process as well.
Short answer, yes. But I'm excited because with this phase, at least for a number of the hits, the curves looked good. So yeah, after going cross-eyed looking at a bunch of the curves, yeah, I mean there are a few that it was trying really hard to give you a number, but overall, I'm pretty confident in what we're calling a selectively active hit.
Yeah, appreciate that.
Yep.
Thank you. And sorry, this is Monique Perrin. I forgot to state my name at the beginning. I'm just breaking all the rules here. Sorry.
Okay, Dr. Berg, thank you for using the raise hand feature. Go ahead.
Thanks for the presentation. Just clarifying question. On one of the slides, it said, did it say 99% of assays, no, 99% of compounds showed cytotoxicity?
No, activity.
Activity.
Yeah, and
got it.
A few of them. I mean, there is cytotoxicity.
At the higher
The higher doses.
Okay.
The cytotoxicity evaluated in the blood.
Thank you.
No, it was active just in those endpoints, so we didn't. I would be interested on that note to look at something on violation. No. Can I? Yeah. So the question. Make sure I got it right. But one of the active compounds in this in the phase 1 was a statin drug used for cardiovascular disease, and that's important in the regulation of cholesterol production, and whether that was taken into account when we called it a hit in the assay. Its mechanism of action in cholesterol. No, our assay endpoints don't model cholesterol or those type of endpoints in the battery as it is. But it would be interesting to look at that, and we've been thinking about a follow-up study in a different assay that may incorporate myelination in testing that compound.
Thank you. Thank you. I am told that this microphone is clicking.
Hey, 1, 2.
But... Okay, so we're having slight technical difficulties with our table mic, so hopefully you can hear online with this microphone. We don't have any more clarifying questions from ICVAM or SICATA members at this time, so we're going to go on to our next speaker. You can go to Megan Culbreath is going to talk about considerations and challenges associated with the transfer and implementation of select DNT in vitro battery assays in a new laboratory.
Does it work?
Pointer forward, backwards.
Right.
Pointer forward, backwards.
Thank you. All right, so hopefully I'll keep this short and sweet for everyone since I stand between you all and lunch. So good afternoon-ish. I want to reintroduce myself actually because although I'm not new to the DNT in vitro space, FDA is somewhat new to the in vitro DNT space. So I'm Megan Colbreth. I'm a supervisory toxicologist and the developmental neurotoxicology program lead in the Division of Toxicology for the Center for Food Safety and Applied Nutrition at the FDA. And today I'm going to discuss our efforts to transfer and evaluate performance compounds in the DMT in vitro battery. So, of course, before I get started, though, a disclaimer, the views expressed in this presentation are my own and do not reflect FDA policy. So, as you all are now aware, several steps were outlined to progress the OECD initial recommendations on the DMT-IBD to an official guidance document. And one of these steps was the transfer of the assays to new laboratories. FDA CISSAN has started the transfer of the DMT-IBD assays developed at the US EPA and IUS. And I want to emphasize here that this is not a direct technology transfer. And for this reason, we have encountered some challenges in this process. However, before I discuss those challenges, I do want to refresh your memory because you obviously haven't seen this table enough today. So on the left, outlined in gold, are the fundamental neurodevelopmental processes covered by the DMT-IBD, and the individual assays are then color-coded by institution on the right. This has been brought up as a question earlier, so I'm going to point out again that most of the cell models employed in the EPA and IUS assays are human embryonic or fetal derived. However, as has been noted, there are only 3 processes not currently covered by a human-derived model, and as Tim mentioned, those are from rodent cells. So, about that. Here I have added, and the colors don't look great, but you can hopefully imagine. Here I have added the in-progress assay transfers at FDA CISSAN. And again, a reminder, this is not a direct technology transfer. So one of the primary challenges we have faced is cell model selection. Of the cell models utilized in the EPA and IUS assays, only one is commercially available. Therefore, we've had to select new models for most of the assays. It's important to note that the cell model employed can have a significant impact on the assay performance. So I'm not going to provide details on all the new cell models selected, but I do want to point out again that we have had to choose new cell models for almost every neurodevelopmental process and associated assay. For today's presentation, however, I will discuss the one assay in which the cell model was commercially available, and I've pointed to it there. This model is the iCell gluteneuron. It's a human-induced pluripotent stem cell-derived glutamatergic neuron, and it's used in the HN initiation human neuroid outgrowth assay developed at EPA. I also want to mention here that for FDA purposes, we are making efforts to adapt every model for human-induced pluripotent stem cells, so every neurodevelopmental process for human-induced pluripotent stem cells. So for the neuroid outgrowth assay, FDA CISSAN employs the same cell model as mentioned and follows a similar cell culture and chemical exposure schema as EPA. Briefly, the cells are seeded in 96 cell plates directly from thaw and allowed to attach for 2 hours prior to exposure. And then 48 hours after plating, cells are fixed and immunostained for high content imaging. I want to note here that FDA CISSAN utilizes a different high content imaging microscope than EPA. Our microscope is a confocal microscope, while EPA's is epifluorescence based. Regardless, similar cell markers are applied in each assay. So it's difficult to see here, but hopefully you can see online. But briefly, DAPI, which is pseudocolored blue in the left image, is used to visualize cell nuclei, and beta-3 tubulin, pseudocolored red, in the right image to visualize cell soma and neurites. FDA SysSan and EPA also assess similar endpoints listed on, which are listed on the right for potential effects on neuroid outgrowth. However, again, different image analysis algorithms specific to the high content imaging technology are applied at each institution. So for brevity in the presentation, I'm not going to provide extensive detail on our concentration response modeling pipeline. However, I do want to note this pipeline is akin to the EPA Toxicological pipeline, which is suggested in the OECD initial recommendations. Briefly, data are normalized within individual plates. Responses are expressed as percent activity relative to the raw median of the vehicle control, plus the two lowest concentrations tested of each chemical. Normalized data are then aggregated across experimental replicates and the 2CP-Alpha2R package applied for concentration response modeling. Importantly, and Chris alluded to this, quality control of raw and normalized data is assessed for all experiments. I do want to quickly take a look at one example. This is for cadmium chloride. So concentration response curves for each assay endpoint are presented at the top. The cutoff or response threshold was set as 30%, which is a pretty universal response threshold set within the Toxicological database for the NT data specifically. And this cutoff is displayed as the dotted gray line on each curve. The hit call from the curves as well as the table below is the continuous activity hit call. And for those that aren't familiar, if the continuous activity hit call is greater than 0.9, this is considered active. The benchmark dose, or BMD or BMC, as people have been referring to it today, it's highlighted in green. This is the concentration at the benchmark response, which in this case is 1.349 times the control standard deviation. Finally, the AC50 is the concentration at 50% maximal activity. And you probably see the ACC there. I will go into more detail about the ACC on the next slide. So for this example, we then compared our results to that of EPA. But first, I want to note that there is currently no consensus on how to compare data across laboratories, and I think that's been emphasized throughout the presentations today. So for the EPA data, these were obtained from the Comtox Chemicals Dashboard, version 2.4.1. So the hit call, BMD, and AC50 are as described from the previous slide. For the ACC, which is the concentration at a defined threshold, as I mentioned, we set our response threshold at 30% activity for all our endpoints. However, I do want to point out for branch point count, the response threshold was 3 times the baseline median absolute deviation, or 4.38% for the EPA data. So for all endpoints except neuron counts, effect concentrations were similar between the FDA SysSan and EPA data, but I will discuss the lack of effect on neuron counts that we at FDA observed later in the presentation. I don't think it's switching. Oh, there it goes. Okay. Next, we evaluated a set of assay developer recommended performance compounds. And just to provide some clarity here, this is seen as a training set of compounds. This is not a commentary on the positivity or negativity of any of these compounds as developmental neurotoxicants. These are just compounds that were expected to be active or inactive in the assay. So although many of these compounds were recommended in the OECD initial recommendations, most either had no data or data for a different cell type on the Comtox Chemicals Dashboard. But fortunately for our purposes, EPA was kind enough to provide the raw data from the original publications for several of these compounds. We were then able to process that data through the FDA SysSan concentration response modeling pipeline so we could compare. So again, for brevity, I'm only going to present data for one endpoint, in this case, neurite length. And as I mentioned, there's 4 endpoints that we collect from this assay, but we'll just look at neurite length as an example. In addition, I'm only going to report the continuous hit call here and the ACC for the performance compounds. Again, in this case, the response threshold was set as 30% activity for both the FDA CISPAN and EPA data. So for this first set of compounds, there was good agreement between FDA CISPAN and EPA data. For this latter set, however, FDA CISPAN discerned more active compounds. So sodium orthovanadate, transretinoic acid, and U0126 were active in the FDA CISPAN HN initiation assay, but not EPA's. And again, I want to remind you that these 3 compounds were recommended as expected actives for neurite outgrowth. So importantly, expected inactive compounds were not active in both FDA CISPAN and EPA assays. So to reiterate, for this assay transfer, the same cell model and roughly the same experimental design were employed. Furthermore, a concentration response modeling pipeline akin to the EPA Toxicological pipeline was applied. As I mentioned, it was a challenge initially to identify performance compound data for comparison. However, in our case, the assay developer provided needed data to overcome this challenge. Again, effects were comparable across institutions for most endpoints. Although for neuron count, which is the cell viability metric, FDA CISPAN was unable to reliably detect effects. Therefore, we have elected to run a separate cell viability assay for this endpoint. So this is again to remind you of the in-progress assay transfers at FDA CISPAN. And for those with a green star in the upper right corner, a performance compound set has already been evaluated. I did not touch on this extensively today, but selection of performance compounds or training sets has been another significant challenge. Often recommended compounds for each assay lack appropriate data for comparison purposes. So obviously our next steps are to continue the transfer of an evaluation of performance compounds in the DNT-IBB. This includes refinement of our concentration response modeling pipeline. Furthermore, we intend to implement new approaches already in development, particularly the 2 listed here. So with that, I'll thank you for your time. And I do want to acknowledge the DNT program researchers who are responsible for the data I presented here today. I also want to thank our colleagues at the EPA. Thank you so much.
Thank you, Megan. At this time, we will be opening it up to clarifying questions from ICFAM and CICATM members. Meng Hang, I believe you have a question. You should be able to unmute yourself, Meng Hang.
Oh, okay. All right. Can you hear me?
Yes.
Yes.
Okay, great. Yeah, because I don't know how to unmute. Okay, so when you run the neurite autogrowth assay, have you tried to use like the cell viability assay along with the neurite autogrowth assay? Because we found some of the compounds actually they kill the neurons. It's not because they decrease the neurite autogrowth, because actually they just kill the cells. So have you, when you run those type of the assay, have you tried the cell viability
Yeah.
assay?
Yes. So that data is coming off or coming out of the lab as we speak. So we're actually running the third replicate of our cell viability assay. It's a cell titer glow assay. So the reason we initially approached it or using an endpoint from the high content imaging was because EPA uses neuron count, which they obtain from their high content imaging as their viability endpoint for this particular assay. We did start with that and we could not detect effects. So we do run a cell viability assay simultaneous with this, this high content imaging approach. And yes, you are correct. Several of these compounds are cytotoxic. I didn't show the data here today, but we look at selective activity similar to what Chris described in his previous presentation.
Yeah, I think it's good, you know, when we use the cell viability assay, there are several cell viability assays. So cell titer glow assay, actually, it's a very sensitive assays compared to the others. So when we run the neurite autoglucose assay, we also face, you know, which cell viability assay you should use. And the cell counting, definitely we will use the cell counting when we measure the neurite autoglucose. But however, the cell counting actually is not very sensitive. What that means, when the cell is dying, you still have the cell, you know, you will see under the microscope, the cell is still there, but they are dying. So I feel, you know, for the DNT panel discussion, you know, when, you know, for OECD guideline, I feel for the cell viability assay, we definitely need to think about what kind of cell viability assay we should use. Okay, so some of the assays are very sensitive and some of the assays not that sensitive. So we want to maybe pick in the middle round or something. So, you know, so that is because when we run the neurite autoglucose assay in the lab, we face like that kind of problem. You know, sometimes we feel the compounds, they decrease the neurite autoglucose, but they also kill the cell. But we can compare IC50 shift, right? We will see like a tenfold difference. But however, some of the compound like the lutein, that compound, if you use the cell titer glow, sometimes, you know, they just kill cells. Okay, then will be inhibit the intracellular ATP content. Okay, thank you.
Okay, thank you.
Question, as you're developing the concentration response models, are you also looking at the human relevant exposure concentrations?
So not currently, no, since we're looking at performance compounds. Again, just to emphasize, these are compounds we expect to be active based on existing data in these assays. We're not speculating on the human relevance of the compounds for developmental neurotoxicity. As we start to look at compounds of interest to FDA, that's when we would move towards that.
Okay, I don't believe that we have any more clarifying questions at this time. So thank you, Dr. Kulbreath. We are going to move on to the public comments session. And just a heads up that after this session, we will be breaking for lunch. So we're going to change the agenda slightly. Okay, so public comments were solicited through the Federal Register notice. Written comments were posted to the meeting webpage and distributed to CICATA members in advance of the meeting. Individuals requesting to make oral comments were required to register in advance for planning purposes. I would like to indicate that there was one written comment from the Humane Society of the United States. And I would like to introduce the oral presenter that we have at this time, who is Randy Ashton from Neurocenter. you can go ahead. The presentation will be remote.
Hi, can you hear me?
We
Hello?
can.
Awesome.
We
Yeah,
can
thank
hear
you. Okay. Thanks for the opportunity to present here. I just wanted to provide an update on our platform that we are commercializing here at NeuroZeta. So I've spoken at these meetings before, and the platform for quantitative high throughput screening of human developmental neurotoxicity. And we call it the Rosetta Ray platform. And just to give a little context, maybe a different way, I don't know if I can advance the slides here. I can't because I'm not presenting. Can you advance the slide? Great. Actually, so another way of looking at the DNT in vitro battery is to look at actually the processes that are being modeled based on the cell types that are being used. And across the top here, you have the sort of characterization of a timeline for human neurodevelopment. And this is from conception through birth. And you can see that a lot of these battery assays essentially come into play after barriers have been put into place that really walls off the central nervous system in development. So this would be the blood-brain barrier that occurs within the 6th to 7th or 8th week of development, as well as the placental barrier. And so there's always a question when you're screening compounds as to whether these compounds actually get to the central nervous system to have the effect that we're screening for. Can you advance one, please? And what's now known from science is that the initiation of what I call flaggable events, whether you're going to have a neural tube defect, a congenital birth defect such as spina bifida, or whether you have the initiation of autism spectrum disorder, this really occurs very early in human neurodevelopment. Advance again, please. And so we have technologies that can model the entire sort of morphogenesis process of the central nervous system, at least for central nervous system tissues. And those are organoid models. Advance one, please. However, these have issues with reproducibility and scalability. And what we've done at NeuroZeta, advance one, is we have generated our Rosetta Ray platform, which really standardizes the generation of early neural organoid formation, forebrain and spinal cord morphogenesis, covering this critical window prior to innate barriers to wall off the central nervous system, and that correlate with the initiation of neurotube defect and autism spectrum disorder pathogenesis. Advance, please. And so our solution at NeuroZeta is to bioengineer human neural organ morphogenesis, particularly looking at the formation of the neural tube structure, which you can see in the top half of this slide. Formation of all brain, retinal, and spinal tissues comes from this structure. And if you take a transverse slice of it, it looks like essentially a tube, an annular tube of neural stem cells indicated in the blue, and they polarize to give a central lumen, which will become your ventricles and spinal canal, and are bordered by extracellular matrix proteins, which again, wall off the central nervous system. Now, for over 2 decades in vitro culture, we've been able to generate these cell cultures and differentiate them into neural tissues. And they go through this same process when you generate these neural stem cells, these early neural tube cells, of forming this rosette structure. But it's random in both space and time and has limited our ability to use this as a actual marker for quantitative analysis. Advance, please. So what we've done at NeuroZeta is integrated this process of generating these neural organoids with micropattern culture substrates, and that gives us a very standardized in both space and time and reproducible format for generating these single rosette structures. And just like you use a mouse and look at its anatomical structure in order to infer some developmental neurotoxicity, here we can basically detect when these single polarized rosette tissues modeling this neural tube-like structure are actually formed. Can you advance forward? And while I'm showing you, I don't think the video would show here, these are 3-dimensional tissues. It would have a nice 3-dimensional diagram here that shows up where the polarized structure that you're looking at is in the middle of these neural organoids that are microarrayed within our well plate format. Advance, please. So what we've done at NeuroZeta is made this into a screening pipeline where we have human pluripotent or IPS cell lines that we can seed from thaw into our Rosetta Ray plates that we manufacture. And we can then expose these tissues as they are growing through this morphogenesis process to various chemical factors. Advance one, please. This gives us both, sorry about the formatting here, that seems to be disrupted, but this gives us the ability to look at forebrain and spinal tissues. They grow over the course of several days, up to a week, and therefore this allows us to basically model the morphogenesis process, both differentiation, proliferation, and formation of these rosette tissues. Can you advance one, please? In a single well plate, we have multiple compounds on the plate. We do full 8-point dose responses over 7 orders of magnitude that we're analyzing concentrations. And at the end of that, we get arrays of these rosette tissues, which we can go back in and image. We run those images through our AI-based software detection process, and that then spits out the data for the endpoints that you see here: cell viability, proliferation, neurodifferentiation, and rosette formation. We also have another data point, which is really a cytotoxicity metric, which is the number of tissues per well. And over the course of several days, if something is cytotoxic, it really does kill all the tissues in the well. Advance 3 times, or advance forward, please. So what this gives us is unprecedented statistical analysis. We can get some mechanistic insight based on the juxtaposition of these curves as to which groups of pathways are being affected. And advance one more time. And because of this, we can really scale this process, being one of the first sort of organoid morphogenesis assays that is now a scalable pipeline that can be used for screening applications. Advance. So what we've done with this assay is quite a bit. That's in this bioRxiv preprint that is now online. We've shown that it can be used for screening, working with DMSO solvated compounds. Advance, please. It has really excellent reproducibility with a Z-factor score of over 0.5 across more than 20 assays. Advance, please. We can do this with forebrain and spinal rosette arrays, really covering aspects of the CNS that you don't cover with the DNT in vitro battery. And we can integrate human metabolism into this. So we can metabolize compounds before adding them to our assay. It is tolerant of those proteins for metabolism. Advance, please. We've also tied this to unique profiles that we see for chemicals, whether you're looking at the forebrain versus the spinal cord. And advance twice, please. And we've used this now in both our preprint and a prior publication to show that we can use it to model genetic factors. So these are gene-edited cells or patient iPSC lines for spinal bifida, as well as autism spectrum disorder. So we are able to model those genetic flaggable endpoints with our assay, further contributing to the biological relevance of our assay. Advance. So we are now in the process of screening compounds, and these are some of the compounds that you've seen in prior presentations. What we've done here for these sort of reference compounds is to show how they overlap with some of the DNT, IVB, AC50s that you see on the graphs here. And really, we can have really good detection across the 7 orders of magnitude that we're assaying to generate really nice dose response curves for our endpoints in the assay. Advance, please. We are currently finishing up a project with the EPA where we're doing a blinded screen of their ToxCast compounds. I just want to highlight that this overlaps pretty much 100% with compounds in the OECD guideline that have been tested across a number of assays. And we have a few compounds that we provided at Neurozetta when we were sort of just getting this up and running. Can you advance one, please? So far, we've gone through about 50 compounds. Our sensitivity is 87%, so really good sensitivity. And our specificity is 100%, but that comes with the caveat that we don't have a ton of negative controls. We are working to increase that and hopefully working with DTT to get some blinded compounds to further increase our negative controls as well. Advance, please. And so I just wanted to provide this update on where we're at in this process. These publications will start coming out with the Rosette Array. We do contract human DNT screening services and are available to collaborate with regulatory agencies in terms of providing this endpoint, this critical endpoint that we see for early neurodevelopment at a time when these barriers natural innate barriers for the CNS are not in place and that correlate with flaggable epidemiological metrics such as congenital birth defects, as well as diagnosis of autism spectrum disorder. We can provide the plates as well as access to our image analysis platform if people want to bring this in-house. And of course, we also can do personalized disease modeling and drug discovery. And with that, I can take any questions.
Thank you. I would like to open it up to clarifying questions from ICVAM and Cicata members at this time. If we can use the raise hand feature. Okay, great. Dr. Bennon.
Can you hear me from there? Okay. So I want to thank all the presenters. I have 3 questions and comments, and it's kind of just clarifying questions. Okay, so we're not open. Oh, I'm sorry then. I misunderstood then. I'll wait for when you open up. Okay.
Sorry. I just wanted to say that it's for clarifying questions only, and we'll open it up after lunch for the discussion portion. So I do not believe that we have any clarifying questions for Randy at this time. Thank you very much. And with that, we are going to break for lunch. And we will return promptly at 1:15. So again, if you want to join a few minutes early, make sure that we can promptly join up back, reconvene at 1:15. Thank you very much.